Drugs for COVID-19. Medicines have been administered to prevent COVID-19. Those medicines are vaccines. They have been also administered to treat mild disease in order to prevent disease progression in high-risk populations, and also to treat severe disease. There is one medicine also authorized for the prevention. In addition to vaccines, it is monoclonal antibody. Now, you see a screenshot coming from a website of European Commission. Here you have medicinal products authorized by European Commission for use in European Union. And all of them are included into today's discussion. First of all, we start with medicines administered for the prevention of COVID-19. Those are vaccines. And this is a list, a full list of vaccines authorized in European Union. We can classify vaccines to three classes in accordance to the mechanism of action. We have ribonucleic acid vaccines. They contain nucleoside-modified mRNA in lipid nanoparticles. Lipid nanoparticles are necessary for entering mRNA the cells, and then in the cell this mRNA direct the expression of COVID-19 as antigen. Then also vaccines containing viral vectors. Vaxzevria contains vector of chimpanzee, and vaccine Janssen human vector, human adenovirus vector. So in both cases, adenovirus has gene of COVID-19 as antigen, and such virus is called viral vector. Finally, Nuvaxovid is all technology drug which just contains as antigen of COVID-19 and an adjuvant which increases immune response. S antigen comes from the word spike. Second step, in case of mRNA vaccines, it is coding of S antigen of COVID-19 virus. It is the same in case of mRNA and viral vector vaccines, and there is no this step in case of Nuvaxovid because contains S antigen itself. Final step in the mechanism of action is in all cases the same, as antigen stimulates neutralizing antibody and cellular immune responses. This is a slide which integrates mechanisms of action of all vaccines. So first we received the nucleic acid vaccines, ribonucleic acid vaccines, then viral vectors, and finally a protein-based vaccine. How do they work in this picture? First, we have this circle in green. It is the same for natural viruses and as well as viral vectors. They enter the body after binding ACE2 receptor, angiotensin converting enzyme 2 receptors. Then they enter the cell once again. This is the same as for natural viruses. They form vesicles following fusion of virus, and then ribonucleic acid is released. Then we have common pathway with mRNA vaccines because, as you remember, it is mRNA with in lipid nanoparticles which allow them to enter cells, and they code the same as protein. Then there is release of spike protein. Spike protein, in case of vaccines, is ingested by antigen-presenting cells. They display this S antigen to T helper cells, which enable other immune responses, including B cells, to make antibodies, and cytotoxic T cells to destroy infected cells. Also, we have antibody treatments available, which will be discussed later. A part of everything what was mentioned, after infection and maybe after vaccination, there is long-lived memory of B and T cells that recognize the virus and can patrol the body for months or years, providing immunity. Therefore, if we base our idea on protection only on antibody titers, this is not scientific view, especially following the infection. Once again, long-lived memory B and T cells following new contact may soon activate and produce both antibody response and cytotoxic C cells, which may protect against infection. Finally, we got a hierarchy of clinical evidence in the evidence-based medicine. We have studies here. There are randomized controlled trials on the top of the pyramid. And they have been called to be gold standard in medicine. We'll have discussion on mostly on randomized controlled trials later, with very few slides included for illustratory purposes. And once again, these trials provide best, most reliable data. Also, we can have cohort studies. This is not the case for COVID treatments, but still, They are also reliable, prospective. They are prospective. So let's say today we form several cohorts. We observe it for outcome. Example could be million women study performed decades ago. Some women took hormonal replacement therapy and others didn't, and they were observed for various safety and efficacy outcomes such as cardiovascular risk of cancer, etc. Also, there are case control studies. They are retrospective. It means that we look at registers. Let's say we look at registers of COVID-19 cases and look at 28 or 60-day mortality following a positive test. We can also make the comparison of, let's say, deaths following vaccination, also making case control studies. And finally, on the bottom, we have here actually not studies, just case reports or case series. Let's say we see a lot of cases in hospitals and reported cases, also narrative reviews. They look at specific problem and review it, expert opinions, etc. So this is at bottom level. And sometimes it may happen that data of various level is not consistent. So in this case, once again, this pyramid is highly important to make sure which data is mostly reliable and which is just for consideration. Efficacy and safety of vaccines. I will use only community to reduce volume of information. Community, as you know, is most often used vaccine. So primary endpoint in randomized clinical trial was protection against symptomatic disease, which was defined as first COVID-19 occurrence from 7 days after dose 2 in participants without evidence of prior COVID-19 infection. So this means that we have no data on participants with evidence of prior COVID-19 infection. And then you see definition of confirmed case. So it is a positive laboratory test at least one symptom of the following. For example, fever or chills or sore throat, in addition to positive laboratory tests. And these are results. These are updated results approximately in the middle of 2021. They show more than 90% vaccine efficacy of in terms of relative risk reduction in all participants. There are little differences for various age groups. Not so reliable data in oldest age group because of low number of cases. Let's have a look. What does it mean 91.3%? So here I just put cases from the table in the website of European Medicines Agency to the Excel table, the same numbers of cases. This is vaccine group, this is placebo group, cases, participants, and then this is percentage of subjects which got COVID-19. So this means that 12.9% of participants were protected from symptomatic disease by vaccine, and we get a relative risk reduction of 91% if we divide this 12.9 to 14.2. To get precise number, of course, we need not only decimals but also centimals. Then secondary endpoint in randomized clinical trial was first occurrence of severe COVID-19 in participants without evidence of prior COVID-19 infection. Once again, in participants without evidence of prior COVID-19 infection. Once again, no data on participants with evidence of prior COVID-19 infection. And then definition of severe COVID-19. So once again, it is confirmed COVID-19 as referred before, but also at least one of the following signs or symptoms, such as severe systemic illness, respiratory failure, shock, significant acute renal, hepatic, or neurological dysfunction, admission to an intensive care unit, or death, and that's it. It is full list of criteria of severe COVID-19. I would say that this endpoint is much more important in comparison with primary one, because it provides really important benefit of vaccine. Now let's have a look on the table on protection against severe disease. Quite high percentage, 96, but unfortunately missing information in which age groups did they find this protection. Just this is aggregated data. And then I put once again this data to the Excel. So we got 0.35% absolute risk reduction. So 0.35% of subjects, of trial subjects, were protected from against severe disease by vaccine, and this makes finally a relative risk reduction of more than 95%. Once again, we divide absolute risk reduction to percentage of cases in placebo group. Unfortunately, Commission drug information approved by European Commission does not contain data on protection against death. However, I found this data in highly respected medicinal journal, the British Medical Journal, and these are all cases of deaths in Comirnaty clinical trial from dose 1 to unblinding. So BNT 162, it was named for Comirnaty vaccine in the clinical trial. Actually, not was, it is, because clinical trial will continue until 2023. And let's have a look. COVID deaths, 2 deaths in placebo group and 1 death in vaccine group. No conclusions can be made because of low number of cases. Then cardiovascular deaths, we got 9 deaths in vaccine group and only 6 deaths in placebo groups. And finally, total number of deaths is 15 in vaccine group and 14 in placebo group. So we cannot say anything about changes in risk of death in accordance to the data of a randomized clinical trial. Then next important factor is protection against transmission, but it is not investigated in randomized clinical trials. This is really a highly complicated issue, because to investigate this challenge is necessary. Let's say we challenge vaccinated and unvaccinated people with infection and investigate then possibility of transmission from vaccinated and unvaccinated people to others. It could be unethical, and this is not, it was not investigated once again. And of course, protection against everything, including symptomatic disease, severe disease, and transmission, changes with appearance of viral variants. For example, we remember that vaccines provide indeed good protection against wild type virus as well as alpha type, but it was less in case of Delta variant and probably even less in case of Omicron variant. And finally, we got adverse reactions in this slide. On the left side, there are very common but transient adverse reactions which last just several hours or days. They are not dangerous, but as you see, they appear in almost all study subjects. There are also serious adverse reactions which are rare or very rare, but frequency of some of them is unknown. It is really difficult to estimate frequency if we have only post-authorization data. Risk-benefit ratio. To estimate this, we should put risk and benefit as close as possible to each other for comparison. So we compare risk-benefit ratio in accordance to protection against symptomatic disease versus mild adverse reactions. Initially, at least until appearance of Delta variant, the vaccines provided quite good protection against symptomatic disease, and mild adverse reaction in most of vaccines. But I think that everybody will agree that it is not best estimation of risk-benefit ratio, because neither protection against symptomatic disease nor acute mild adverse reactions are important problem in medicine. Then let's have a look on protection against severe disease. We got this data in the lecture. So 0.35% of patients, of subjects avoided severe disease because of vaccines. But also there is a risk of serious adverse reactions, both short-term, which were discussed in previous slides, and also long-term, which may be unknown. So this comparison is quite difficult at the moment, and probably conclusion may be related to concomitant diseases, to age, etc. So risk-benefit ratio may be different in young, healthy people and elderly people with concomitant diseases. Also, if we have a look on the protocol of the randomized clinical trials, we'll find which subjects were not included into them. So this means that we have no highest level scientific data for these subjects, and this is a list of example. It is not exhaustive list. For full list, you should refer the protocol. The link is below. So once again, there is no data in subjects with evidence of previous COVID-19 infection, no data during pregnancy and lactation. Yes, we have some post-authorization data. Unfortunately, also in clinical trials, no data in subjects with immunosuppression, and even women with childbearing potential not using acceptable contraception were excluded from randomized clinical trial. And then we go back to protection against severe disease. We have just aggregated data without information on specific high-risk groups or age groups. Also, if we have a look on booster dose, we know that it was authorized by European Commission in 2021 in October. So they allowed it to use at least 6 months after second dose. It is not a recommendation. It comes in the annex to the decision of European Commission, so it is legally binding. And the Commission advises, based on opinion of European Medicines Agency, that decision when and whom to implement the third dose should be made based on vaccine effectiveness data and taking into account limited safety data. You probably noticed that in the lecture I speak about efficacy. Efficacy term applies on clinical trials which are performed on ideal conditions. So clear-cut inclusion-exclusion criteria, monitoring of patients, etc. Effectiveness is a little bit different parameter, which means data which we collect in practical conditions. So it is post-authorization surveillance data, not highest level data, but still they recommend to use it. Unfortunately, safety data of booster dose is limited. And what is also important, effectiveness of a booster dose is based only on neutralizing antibody titers, which is really disappointing because what is most important is clinical outcomes, especially protection against death and severe disease. And we have no data of clinical trials. And also, unfortunately, even data on antibody was gathered in individuals from 18 to 55 years of age. So no data on ages which are more susceptible to COVID-19 disease, and therefore who probably need vaccines much more than younger and healthy people. Medicines for the treatment of COVID-19. They have been used to treat non-severe disease. It means to prevent disease progression and also to treat severe disease. And on this slide, you see all medicines authorized by European Commission, also including vaccines. In addition, we have dexamethasone, which have been used for the treatment of severe disease. It is authorized nationally. In addition to other indications, it is also authorized for COVID-19. Medicines used to treat non-severe disease include monoclonal antibodies specifically designed for COVID-19 and repurposed antiviral drug remdesivir. All monoclonal antibodies have comparable mechanism of action. They bind as antigen of COVID-19 virus and block its entry to the cells as it was explained in the section on the mechanism of action. In addition to the treatment of non-severe disease, casirivimab-imdevimab combination is indicated for the prevention of COVID-19 in selected patients. Here, next slide, it is inclusion criteria to the clinical trial. So this is target populations, unvaccinated people with at least one risk factor. And the table shows clinical data on primary endpoint, which was COVID-19 related hospitalization or death through day 29. Below, there are absolute risk reduction and relative risk reduction for both doses. I just remind how these percentages have been calculated. So if we have 3.4% of trial subjects with events in placebo group and 0.9 in investigatory drug group. So we subtract 0.9 from 3.4 and get absolute risk reduction. Then this means that those 2.5% were protected by drug. And if we divide 2.5 by 3.4, then we receive relative risk reduction. Regdenvimab is also monoclonal antibody indicated in similar populations Also, we have absolute risk reduction and relative risk reduction. Just primary endpoint is a little bit different. Also, inclusion criteria and trial centers are different, so comparison is not possible, but of course we cannot make any conclusion on which drug is better or which is worse in accordance to this data. And finally, we got third monoclonal antibody, sotrovimab, once again with similar indication, similar mechanism of action, and similar efficacy, both in terms of primary endpoint, which was progression of COVID-19, and definition is here. Also, secondary endpoint progression to severe or critical respiratory COVID-19. This is relative risk reduction. If you want absolute risk reduction, it is 5% here and 4% here. So once again, comparable percentages. I would say that once again, there is no data demonstrating that any of antibodies is better than another. Finally, we have remdesivir. It is adenosine nucleotide prodrug. It has similarities in mechanism of action with some cytotoxic drugs, antimetabolites, and also some antimicrobials. It is metabolized to remdesivir triphosphate, which competes with natural adenosine triphosphate for incorporation into ribonucleic acid, and is used in patients both with non-severe COVID-19 and severe COVID-19. Indication of non-severe COVID-19 has been authorized by European Commission in the beginning of 2022. So this is inclusion criteria to the trial of the treatment of non-severe disease, and data on primary endpoint. Once again, comparable efficacy with monoclonal antibodies as discussed earlier. Finally, we have medicines used to treat severe disease, the same remdesivir may be used in hospitalized adult patients with COVID-19 with evidence of lower respiratory tract involvement. And on the bottom of the slide, you see analysis of post hoc, and it means not intended, not designed before trial, but performed later, post hoc analysis of 29-day mortality. And you see great differences in two groups: patients who required low flow oxygen benefited largely. It is more or less relative risk reduction is approximately 3 times. Meanwhile, benefit was not found in patients who required high oxygen or high flow oxygen or non-invasive mechanical ventilation. Finally, tocilizumab, monoclonal antibody. It binds to interleukin-6 receptors. Interleukin-6 is pro-inflammatory cytokine involved in T-cell activation and induction of immunoglobulin secretion. So it has been used for the patient in whom immune response is too strong and cause harmful effects on the body. So it is drug only for severe disease, not investigated in other patients. So in adults who receive systemic corticosteroids and require supplemental oxygen or mechanical ventilation. And there is data on the reduction of risk of death. So absolute risk reduction is quite comparable with drugs which we discussed earlier. However, relative risk reduction is much smaller. Now we got other immunosuppressant. It is anakinra. It also inhibits action of interleukin. It inhibits binding of interleukin-1 alpha and 1 beta to interleukin receptor type 1. Explanation of pathophysiological role of interleukin-1. Once again indicated in adults with pneumonia requiring supplemental oxygen, who are at risk of progressing to severe respiratory failure. But endpoint, it is just significant improvement of clinical status, not risk of death. WHO CPS stays for clinical progression scale of World Health Organization, and here there are explanations. Finally, dexamethasone, well known since many years, corticosteroid which has only anti-inflammatory potency, and has been discussed in the section on corticosteroids. It has been used to treat, once again, severe COVID-19. So in patients where additional oxygen is needed. And here there are slides just showing possible mechanism of action. Probably it is dexamethasone may inhibit development of cytokine storm, fluid accumulation, and edema. Pulmonary edema is responsible for respiratory distress. So that's it, and thanks for your attention.